Lieber Peter, vielen Dank für diese Einladung und ich bin sehr geehrt, hier zu sein. Ich freue mich, hier in Deutschland zu sein auf der DWG-Kongress und es ist besonders super, hier mit so vielen Freunden und guten Kollegen zusammen zu sein. So having said that, I will, with your forgiveness, switch over to English for my presentation. And I'll be giving some perspectives on the repair and regeneration of the injured spinal cord. And I'm a fan of the Impressionist paintings, and this lecture has been inspired by some work of Paul Gauguin. And I'm going to give you a sense of where we've been, where are we now, and where are we going. So by way of disclosures, I have spun off a biotech a company that's in the regenerative medicine space. This is being managed at arm's length from me. I've been a PI on a number of clinical trials. I'll refer to some of these, and I've received a number of sources of funding. And again, by way of acknowledgement, many thanks to the organizers of the DWG, and in particular to my dear friend, Professor Peter Vaikoshi, for this honor. So this is where I come from, and I work at the Kreml Neuroscience Center, which is one of the big teaching hospitals at the University of Toronto. The official language in our clinic, of course, is English, but the unofficial language is actually German. It's supposed to be French officially, but we have many wonderful students and colleagues from Central Europe. So this is a painting by Paul Gauguin that I made reference to, and it's entitled in French, "D'où venons-nous, que sommes-nous, où allons-nous?" And I'm sure all of you speak French probably better than I do, but the translation is, where do we come from, what are we, and where are we going? And I really like this concept, and I think about this quite a bit. I'm also inspired by all of the clinical cases that I've managed, and this was a particularly challenging situation because this was a good friend of one of my colleagues, dove into shallow water in one of the beautiful northern Ontario lakes, sustaining the C4-5 fracture dislocation with a C4 Asia A cervical cord injury. This person underwent early surgery. We were able to get some recovery, but not surprisingly, this person is left with a lot of neurological impairment. And so what is our treatment approach in the modern era? Where are we now, and where are we going to be going? So here's where I'm going to provide a few perspectives. So I'm going to be talking about the concept of neuroprotection, and we as surgeons have an important role to play, and this is really time as spine, the role of early decompression, and I think this is continuing to evolve. And what are the emerging strategies in what I might call the post-methylprednisolone era? And we've learned a lot. And I'll give you a little bit of a sense also on how we might be able to use machine learning and artificial intelligence approaches to navigate big data to handle the heterogeneity of outcome prediction. And then I'll be making reference to some neuroregenerative approaches. I'll also be briefly discussing the concepts of bypassing injured neural circuits, and then I'll close out my lecture by making reference again to Paul Gauguin. Where are we now in 2022? And I might speculate where we will be in a decade from now. So time is spine. Early neuroprotection mitigates secondary injury mechanisms, enhances outcomes. And this is a key principle that guides surgical and pharmacological approaches for acute spinal cord injury. And this really forms the backbone to the multi-pronged approach to spinal cord injury care. And there's a very strong rationale to consider the concept of early surgery. And the whole key here is to try to mitigate and to attenuate the events of secondary injury. There is very robust preclinical data to support the efficacy of decompression, and this is beautifully summarized in this meta-analysis by Peter Batchelor from Australia. And across many studies, decompression improved outcomes quite dramatically in animal models. Now, it's one thing to see things in animal models. It's not always so easy to translate this into man. And this really was a major struggle for me over the first portion of my career, and this ultimately culminated in the publication of the Staskis trial. So this was a prospective cohort study in 6 centers, over 300 patients, and in essence, we found that the odds ratio of patients having a major improvement in neurological function, 2 or more Asia grade change, was almost 3-fold higher if patients had surgery at a mean of 14 hours and within this key 24-hour time frame. And so this spawned off the first clinical practice guidelines. These were published 5 years ago, and for the first time it was suggested that early surgery be offered as a preferred option. But we were really only able to make this as an option recommendation, largely because of the inability to undertake a quantitative meta-analysis. And I took this really as a challenge, and this then spawned this publication that came out last year in The Lancet, and this was on a pooled meta-analysis in over 1,000 prospectively accrued patients. So there are 2 graphs here from that paper. The first one on the left, the red line is 24 hours, and these are the neurological outcomes. And you'll see a very steep dose-response relationship between neurological recovery and timing. And the second graph shows the clustering of the outcomes, and you'll see the peak, again, of changes in motor function occur within the first 24 hours. Now, one of the controversies also has been around so-called central cord syndrome, that somehow it's been thought that, oh, well, even though these patients have a severe injury, that somehow they have a wonderful outcome with non-operative management. Well, I can assure you that they actually do not. And so this paper from our group was published earlier this year in JAMA Surgery. This was a prospective propensity-matched cohort-controlled study in 186 patients with central cord injury. And in essence, we found that even in these patients, there was a significant benefit to early surgery. And this was particularly seen in the severe patients, the AIS-C patients. The failure to see an improvement in the AIS-D patients likely reflects the issues that you run into with ceiling effects. And I think one of our challenges is to come up with better and more sensitive outcome measures to look at this in these patients. And this is really these conclusions. And so unless patients are older and frail with medical comorbidities, it's our recommendation that patients have early surgery, even if they have a central cord injury. And then interestingly, right around the same time, in the open access version of JAMA was this randomized trial out of Japan, showing in a different way, but similar results with central cord types of injuries of improved outcomes. So time is spine. So where are we going now? Well, the guidelines that are referred to will be updated, and stay tuned in 2023, and I'll give you a little bit of a preview of what will be influencing those data. And of course, there's a lot of interest now in ultra-early surgical decompression, but we do need the evidence to support these protocols. And then there's different areas of discussion. Should we be doing duroplasty? What's the role of intraoperative ultrasound to assess the decompression? How do we personalize the approach? And these are extremely interesting, important questions. So my colleague Brian Kwan and I are co-leading an effort that's jointly sponsored by AO Spine and the Praxis Institute in Canada, and we have 3 questions that we're looking at. One will be an update on the role and timing of surgery in acute spinal cord injury. The second will be recommendations around the optimal hemodynamic management. And the third is a very controversial area, very challenging, the issue of intraoperative decline in neurological function. So I'll just give you a snippet of some of the new emerging data. So we now are able to derive high-quality meta-analyses and forest plots. And in a nutshell, early surgery within 24 hours is associated with a 3-fold increased likelihood of large changes in neurological function. The hospital length of stay favors early surgery, and there's no change in major morbidity or mortality, and in fact, there are significant trends toward reduction. So early surgery is certainly very safe. So I'll just leave you with those thoughts. Now, one of the challenges we also have in classifying and examining the outcomes in spinal cord injury is how do we deal with the heterogeneity? And we know this from clinical experience that it can be sometimes quite challenging to predict the outcomes, particularly in patients who have an incomplete injury. So this depicts some of the classical methods that we use to assess a neurological examination. And of course, these remain very valid, but they're inadequate. So let me just now show you a graph. So this is from our data set, 801 patients with a cervical incomplete spinal cord injury. And it looks like a snowstorm or like a tsunami. There's no way that you could possibly discern treatment effects with that kind of a graph. So how do we come to terms with this? And this is what we struggle with all the time in randomized controlled trials. So this is where big data approaches can help. So to assemble this, we've been able to put together one of the largest data sets in the world on prospectively accrued SCI cases. And here I'll introduce you to one form of a machine learning approach. This is a supervised machine learning technique called group-based trajectory modeling. And here, what you do is you look at certain clinical variables to try to define what the outcome trajectories will look like. And this has very practical clinical applications. Now, the table is very busy, but I'll highlight a few of the points here. So the Asia grade is certainly important. That is no surprise. The level of the cervical injury is important. You'll see again that there's a clustering of patients who have good and excellent outcomes with early surgical decompression. And you'll also see that central cord injury does not really emerge as a significant factor. And so this is summarizing this again in a little bit of a different way. So looking at the neurological level, looking at the Asia grade, the age is an important factor as well. So again, here is this snowstorm that we're dealing with, but you can now segment the recovery trajectories to define poor, moderate, good, and excellent outcomes. And these are the means and 95% confidence intervals. So you now can actually plot what an individual patient will look like, and you can very accurately model the trajectory. And by the way, the notion that you cannot predict the outcome of a patient in the first 24 hours is nonsense. You actually can. All of these data were collected, very simple clinical data in the first 24 hours. The key is to use a big data approach. And so there's some, I think, very important conclusions and implications here from terms of having prognostic relevance, looking at distinct temporal profiles of recovery, tailoring treatment approaches, and very importantly, risk stratification for clinical trials. And this is a second paper that we published last year in Annals of Neurology, also looking at the variability in complete traumatic spinal cord injury, again, using a similar type of an approach. So what's emerging? And the drugs and the approaches in brackets, minocycline, CSF drainage, magnesium polyethylene glycol were promising approaches. These were studied and did not show significant effects. Hypothermia or cooling is an area that has some interest. The group in Miami and the group in Australia led by Peter Batchelor are examining this, so we'll wait to see. We know that this is a two-edged sword. We know from traumatic brain injury that the mortality rates were higher with systemic cooling. And so I think that this is an interesting area, but I would urge a caution and thought. So I'll talk a little bit about our work with riluzole, and this is a repurposing of a sodium glutamate blocker that is used in clinical practice by neurologists in ALS. So here's the idea that we've been studying. So we've been focusing on cervical cord injuries. Here I'm modeling this as a subaxial cervical burst fracture. So the primary mechanical injury sets in motion that cascade of events known as secondary injury. So here we have the nerve fibers. And at the microstructural level, there are sodium channels. So sodium enters very quickly. There's a reverse operation of the sodium-calcium exchanger and cellular swelling that occurs, and there's a pathological release of glutamate and a failure of glutamate uptake mechanisms. So these are the classic mechanisms of excitotoxic cell injury in secondary injury. And so what we're now studying is the use of riluzole, which is a sodium channel blocker. So we're looking at cervical injuries. The patients are having early surgery, standard of care approaches, and then they're receiving riluzole in an effort to try to attenuate these secondary injury mechanisms. Now, to support the clinical trials, it's important to have robust preclinical data, which certainly were there, but also robust phase 1/2a clinical trials data in men. And this is what we did here. So this is in 72 patients, 36 controls, 36 riluzole subjects, and we found in the patients that received riluzole a significantly higher conversion rate on the Asia Impairment Scale grade, and particularly for cervical injuries, an improved trajectory of motor recovery. So based on this, we launched, and we've now concluded a phase 3 randomized controlled trial. This is currently under analysis, and I anticipate announcing the results of this in 2023. So where are we now with neuroregeneration? Well, this is a chronically injured human spinal cord, a postmortem section. And this is a very hostile cellular environment which limits cell survival. And there are inhibitory molecules that limit axon growth. There's ion dysregulation that leads to aberrant plasticity. So if we want to try to target this kind of a lesion, we need to be thinking outside the box with regard to regenerative approaches. So where are we? So this is actually a Central European story. This is the seminal work of the basic scientist Martin Schwab from Switzerland, who together with Stritt matter from the United States discovered the molecule NOGO. This is one of the key inhibitory molecules expressed on neurons, and it's activated by various molecules in the disrupted myelin sheath. And Armin Kurt, out of the University of Zurich with the EMSCI network, has launched and now completed a phase 2/3 randomized controlled trial looking at monoclonal antibodies targeting NOGO-A. And I anticipate that the results of that work will be available in 2023 as well. This is work that's ongoing right now. There are actually two companies that are sponsoring separately trials. One is AbbVie and one is Mitsubishi Pharmaceuticals. And both companies have developed monoclonal antibodies targeting RGMA, which stands for repulsive guidance molecule A, another inhibitory molecule which is upregulated after spinal cord injury. This is work that has been spun off by the bioengineer at MIT University, Bob Langer. And this is looking at a neurospinal scaffolds. And his spin-off company is called In Vivo Therapeutics. This is a highly porous scaffold. And the idea here is to actually do a myelotomy and introduce this in patients with an Asia A thoracic spinal cord injury. And so In Vivo Therapeutics is currently midway through a phase 2 clinical trial in the United States. And there's a lot of interest in this whole idea of bypassing injured neural circuits through epidural spinal cord stimulation, through advanced rehabilitation strategies, and through brain-spinal cord interfaces. And these are some significant publications from the American group of Harkema and Reggie Edgerton, and here in Central Europe, the amazing work of Grégoire Courtine out of Lausanne. And all of this work is now being translated into clinical trials, and I actually envision the potential for an entirely new specialty in spine surgery, where we have spine surgeons that are dually trained in both spine surgery as well as in functional neurosurgical techniques to be able to try to deliver these types of therapeutic approaches. And then I'll just close off with cellular therapies. And you know, these have been studied in various forms. There's been a lot of hype, which perhaps has exaggerated, you know, some of the effects. But There also is excellent science behind this, and I truly believe that we are about to enter into a new era with what I refer to as next generation stem cells. So I'll briefly discuss neural stem cells, Schwann cells, and endogenous stem cells. So one of the cool concepts is, what if we could actually harness the regenerative potential of endogenous neural stem cells? And this is work actually that our group published over a decade ago in Cell Stem Cell, where we postulated that there was areas in the center of the spinal cord, around the central canal, that harbored endogenous neural stem cells. And this is work that was recently published by Jonas Friesen from the Karolinska in Science, validating what we had postulated. And in fact, what Friesen showed was that they were able to activate latent genetic programs to activate these ependymal cells. And this is work published from our group in Toronto. One of our students, Lorene Hatcham, who actually is a neurosurgery resident doing her PhD in regenerative medicine. And Lorene is examining the idea that a form of ionotropic glutamate receptors called AMPA receptors are actually key to the modulation of these endogenous stem cells. So where are we now with clinical trials in neural stem cells? And I think we're at an inflection point. This is a photograph of me in the operating room. I had a collaboration with Armin Kurt and with Stem Cell Inc. out of California. And we were studying neural stem cells. And we had 2 clinical trials that we had undertaken. One was in thoracic injuries, and then the second was in cervical injuries. And then unfortunately, the cervical injury trial was terminated because of financial difficulties with the company. And this is a challenging area for sure. So our experience here with this has been summarized in the Lancet Neurology. And we showed the feasibility and the safety and the proof of concept, but there were many limitations as well. So were the cell types that we were using the best? What was the viability? How do we really know about the cell fate? And we had limited data in translationally relevant cervical injury models. The techniques that were being used did not consider the perilesional scar, which is a very significant challenge. And I would argue that we were probably using the wrong cells for the wrong phase of injury. For convenience sake, many of the studies were being done in patients with a chronic lesion, using cell types that had been studied in animal models of an acute lesion. And I think that was really a critical error. And in addition, the patients did not receive targeted rehabilitation. They got rehabilitation, of course, but we now know that you need to actually target the different types of rehabilitation to drive tasks. So to move this forward, we developed a high-fidelity model of cervical injury in animal models. Very tricky to do, and we do this with a clip compression contusion model at the C6-7 level. This produces a very severe injury. We can image this. We have beautiful behavioral readouts. And we now can actually study human neural stem cells in animal models using this T-cell deficient road-nude rat. And this has had a dramatic impact on our ability to undertake mechanistic studies of human cells in an animal model. So we've taken 3 approaches to try to improve the effect of neural precursor cell transplantation. So one is to improve the survival, differentiation. The second, to modify the microenvironment, and thirdly, to combine cell therapy with rehabilitation. And this is just what I'm going to just briefly touch on now. So we need to focus on the poor transplant survival. We need to target the cells for the area of the nervous system that we're going to try to repair. And then we need to consider these extracellular matrix changes. And I've called these spinal microenvironment modifying a regenerative therapeutic, or SMART cells. And that was a little bit of a joke on me. And so we are looking at techniques to introduce growth factors, to regionally specify these neural stem cells, and to ways to modify the extracellular matrix. So one approach is to try to introduce growth factors, and this is work that we published in Science Translational Medicine, using a novel technique that we've now spun off into this biotech company I mentioned, where we can engineer these neural stem cells to express a growth factor called glial-derived neurotrophic factor. And this has a dramatic impact on enhancing the survival and differentiation of these neural stem cells. And this is just some figures from this Science Translational Medicine paper showing that we have about a doubling of the survival. Another approach is to try to alter the extracellular matrix. And so this is a self-assembling peptide called QL6. And this comes in a liquid form. Surgically, you can micro-inject this into the injured spinal cord, and this will form a matrix. And then this beautifully integrates into the spinal cord, and we see in the upper curve dramatic improvements in function with this kind of a combinatorial approach with stem cells. Now, one of the challenges with any kind of regenerative therapeutic approaches is to try to get the nerve fibers to actually grow across the lesion. And so to try to address this question, my student Chris Ahuja did an experiment where he used different fluorescent molecules to label engrafted neural stem cells, rostral in green, at the lesion in red, and then caudal to lesion in blue. And so if you look in the upper left, you'll see that there are green nerve fibers which have crossed the lesion. Then you'll also see that the nerve fibers from the lesion epicenter have also crossed over into the lesion. And then finally, we've developed approaches to engineer in molecules to bust the scar. And this is using a CRISPR-Cas9 AAV S1 safe harbor a vector. And we can link this with a red fluorescent protein. And these retain the human neural stem cell characteristics. So this is what this looks like ex vivo in culture. So CSPGs are chondroitin sulfate proteoglycans in the scar. So here's what happens if you introduce conventional neural stem cells next to this scar. It's very difficult to penetrate the scar. And I think this is one of the reasons that the outcomes of conventional neural stem cells in chronic spinal cord injury have been disappointing. But here's what happens if you engineer these cells to express chondroitinase, a scar-busting enzyme. They degrade the scar, and they beautifully cross and infiltrate across this. And then we see very nice improvements in neurological function with various skilled and unskilled forelimb functions. We see in these green-labeled sections that these axons grow for many millimeters beyond the injury site. And then interestingly, they seem to reorient the host astrocytes to be growth permissive. And then finally, another critical factor is examining targeted rehabilitation approaches and looking at the synergistic effect of targeted rehabilitation with cellular transplantation. This is a collaboration with Karim Fouad out of the University of Alberta in Canada, and Karim has developed robots that can actually do targeted rehabilitation, and we can measure this. And then, so we have the animals are trying to reach for these pellets. We can introduce these pellets, and then we can train the animals to recover forelimb function. And this is what the behavioral outcomes will look like. So the lower graph shows the vehicle controls. The middle graphs show improvement with the neural stem cells. But when we add the rehabilitation, we see a dramatic recovery. And targeted rehabilitation was not part of the protocols in the previous neural stem cell trials, which I think was a critical error. And then we've confirmed this electrophysiologically, and we see a beautiful impact in terms of reducing the size of the cavity on anatomical sections. And so I think that we're about to embark in a new era where we're looking at targeted engineered neural stem cells that can either express scar-degrading enzymes, can be introduced synergistically with nanopeptide biomaterials. And also, it's important to have targeted rehabilitation as part of this approach. So just in closing, and coming back to the Paul Gauguin painting, so where are we now in 2022? Well, I would say that time is spine. There's very little discussion around this. The spinal cord injury is a time-critical event. Surgical decompression needs to be done in patients, and it should be done as soon as is medically feasible, and certainly whenever possible within the 24-hour time period, if not sooner. Hemodynamic optimization is here, but is this the correct target in all cases? What's the duration of treatment? And we'll be examining this in the revised guidelines. Steroids are an option, particularly for cervical injuries, but we need better drugs, and I think some neuroprotective options are emerging, and I'll be interested to share with you the results from the Riluzole trials in 2023. Active targeted rehabilitation needs to be part of our protocols, and I think that electrical stimulation to activate neural networks is emerging as an attractive option. So where will we be in a decade from now? Well, I think that early surgical intervention will become the international standard of care. There still is resistance in some areas, and there also are logistical and financial issues as well in low and middle income countries. And what are the optimal techniques? We need personalized approaches to individualized care. And here I think that the whole era of genome biology could have a potentially dramatic impact. We need to really be looking at techniques to assess cord perfusion. The assessment of mean arterial blood pressure is a very rough guide, and I think that there will be advances in this area. And I'm hopeful that we may have some regenerative options for spinal cord injury, which might become available to assist our patients that have very severe residual neurological impairment. Electrical stimulation to modulate neural circuits is the real deal, but it's not going to work in everyone. And we need to be able to personalize these kinds of approaches. And then I think there are many opportunities for we as surgeons to be working together with our rehabilitation colleagues to augment the outcomes for our patients. And on that note, I'll close, and thank you so much.